Welcome to MetPhys 2020, where we do science the inclusive way. That's right, it's open for anybody who wants to watch it anytime, anywhere. It's also cost effective. The only cost incurred here is to the White Megara lab, and we include our kids in the broadcast. Say hi, Sid.
Hi.
We're hoping to have one of the new kittens, but Bruno ran away, right? Yeah.
And so did Aspen.
So did Aspen. All right, they're out of here. But you don't need to fly anywhere to see our kittens. We'll bring them right to your house through Zoom. I'm pretty impressed by Cassidy. She seems to be the only person who's taking this MetPhys 2020 challenge seriously. She's taken a photo of herself in front of the screen every day right now, 16 days in a row. We're ready for a new one. Looks like she slaughtered that scoop monster, and he's wearing his fur in her hair. Well done. Thank you for doing that. Since no one else is posting anything, then I'm going to show you something new that we've got now. The music before the show is now part of a Spotify playlist that you all have access to. I tweeted the link yesterday. Hope you enjoy that. Every morning there'll be new tunes added. The ones from this morning are already added. And hope you enjoy that. We, in our house, are doing the MetPhys 2020 challenges. Martin and I are still working on that dance move. My back has recovered and we're good. Sid and Logan are learning to ride bikes. And Juju is potty training herself. So if we can do all that in our one house, I want to see what you all are doing at home. Come on, let's share some fun to help us get through these days. Fails are as good as successes. We all enjoy those more. I'm sure of it. All right. A few days ago, Joshua asked a really important question. How should we ask questions? Same way he did. We do it through Twitter. We use the hashtag MetPhys2020. If you're not on Twitter, it's free and very easy, and you don't have to have a profile that reflects your name so you can remain inconspicuous. All you have to do is sign up. You can be an egg, and if you go to the search bar and type in the hashtag MetPhys2020, you can follow all the latest stuff that's going on simply by clicking the latest tab. If you also go to the speakers pages, you'll see when people have tweeted them directly and their responses. And I can tell you there's been a lot of insight shared through those question period time. The other nice thing about having our question period on Twitter is there's no limitation. You can ask as many questions as you like and have as many follow-up questions. We're even open to comments that are more of a comment than a question and have nothing to do with the talk, because we can ignore those on Twitter. All right. We've said to remember everyone's faces. These are the early people that very quickly said, we're happy to give a talk and share our data so that all our colleagues can get through this crisis with at least a little bit of metabolic physiology. Yesterday, we heard from Sophie Treffilet. I have some fantastic news for all of you that run institutions and share departments around the world. She is on the job market, so reach out to Sophie. I'm sure she'd be a great faculty member for anyone who's looking. There are additional speakers. It's hard to believe we're already halfway through week 4. We're going out to 6 weeks of talks, and these are the wonderful speakers who agreed to fill out the lineup. I'm sure you'll recognize many of those faces, and if you don't worry, tune in. You're going to hear some fantastic talks. We're going all the way out to April 24. And once again, those talks are thereafter online, both on our Facebook page and on YouTube. I'm also setting up an Instagram page to get the young ones involved. It looks like our metrics so far are 26 and older. I'm assuming the oldest are on Facebook, the medium are on YouTube, and hopefully we can get some young ones excited about metabolic physiology by using another platform. If you'd like to thank me personally, you can do that by supporting the organization that supports me. That's the Pathway to Stop Diabetes program from the American Diabetes Association. You can see the link there. They not only support me, they also support Celine Riera, who's another speaker, I think coming up next week. And we heard yesterday from Sophie how the American Diabetes Association also supports her work, and I'm certain it supports more than just that. So if you would like to say thank you and you have the means to do so, a generous gift to the American Diabetes Association would make us all proud. All right, so without further ado, except to remind you of tomorrow's next speaker, I do that every day. Tomorrow, so we keep asking, are these talks can they get any better? They can, they're gonna keep getting better. And tomorrow is evidence of that. We're gonna hear from Kristen Stanford from OSU about exercise-induced adaptations that improve metabolic health. So all the ex-phys people out there, this is your day, enjoy it. But stay tuned for today as we hear from Thales. I'm going to pass the reins over to him right now, making you host. And you are now host, Thales. Take it away.
All right, so let's see. So, all right, so great. All right, so I can get started. So thanks, Phil, for this amazing opportunity. Who would have thought a month or so ago when we met during my visit to Duke we'd be doing only these online sessions. Our labs would be shut down, and this would be sort of the only source of interactions we would get with other scientists. But it's very important. I've really enjoyed all the talks so far. I've learned quite a lot of new things. So my lab is actually the arrow showing up. Sorry. Okay. My arrow's not showing up, but that's okay. Okay. So yeah, so my lab is in NYU, New York City, which is of course currently under a lot of craziness going on with the coronavirus, but we'll get through it. You can actually see our lab sort of in the background there in one of those buildings on the East River. So my name is Thales Papagiannakopoulos. I also go by as Papa G at NYU. Our lab is interested in understanding what are the different metabolic bottlenecks that occur during tumor evolution, how cells overcome it, and how we can target them therapeutically. So on the left, you have a single epithelial cell, which upon oncogenic transformation by some sort of mutation, so for example, like KRAS or P53, will cause higher proliferation. Eventually, these cells will have to vascularize and form adenomas, eventually adenocarcinomas, and the larger tumors. And as these cells, these cancers undergo this process, they have to overcome certain metabolic bottlenecks that occur, such as nutrient availability, low levels of oxygen, and for example, and reactive oxygen species, which were nicely introduced by Isaac Harris last week in his presentation. So ultimately, the tumors will get, as they get larger, they'll become more heterogeneous, both in terms of the tumor cells themselves. So for example, we have tumor cells with the various mutations, but also in terms of the immune cells that come and are attracted into the tumor. And ultimately, some of these tumor cells that will become metastatic will metastasize and to distant organs. So again, during these different stages of disease progression, during carcinogenesis, there's different metabolic bottlenecks occur, and we really want to understand how tumor cells overcome them and how we can target the mechanism by which to overcome it. And we've done a lot of work with which I've covered today, understanding how the various mutations that occur during tumorigenesis, during progression, can promote progression during these different stages of the disease. But we've also done quite a bit of work, and new work ongoing in the lab, trying to understand how changes in general physiology, so for example, a circadian rhythm disruption, so alteration in the light cycle of a human, and of course we model this in mice, how that impacts tumorigenesis, and we've shown that circadian rhythm disruption can promote cancer. In addition to that, we're trying to understand how diets interact with tumor progression, and how different diets can impact different genetic subtypes of cancer. So we primarily focus on lung cancer. The reason we focus on lung cancer, it's the major cause of death in men and women in the US and worldwide. So despite the decrease in smoking, which is a major cause of cancer for about 70% of patients, it's still a major major issue clinically. In the bottom, you can see a pie chart demonstrating the different histological types of lung cancer. This is a very sort of gross breakdown here, but basically these are the histological subtypes as seen by pathologists. However, lung cancer is a very complicated disease. Even though the tumors histologically may look somewhat similar, genetically they're very diverse. And this is sort of annotated here by just looking at, this is all the tumors ranked, all the tumors sequenced by the Cancer Genome Atlas, TCGA, and then ranked by mutation frequency. And as you can see on the right, along with melanoma, which of course due to UV radiation has a high mutation burden, the two major subtypes of lung cancer, lung adenocarcinoma and lung squamous cancer, have really high mutational burden. So again, this suggests that patients with these tumors have high mutational burden. There's been a lot of sequencing studies trying to characterize what are the different mutations that and the different genetic subtypes in patients. These are the top mutations ranked here by frequency going from top to bottom. As you can see, the top two are p53 and KRAS, two very well-known genes in carcinogenesis. KRAS, a very potent oncogene in lung cancer and other cancer types. And p53, a very important tumor suppressor mutated in approximately 50 to 6% of all cancers. However, the two other most frequently mutated genes after KRAS and p53 are Keap1 and STK11, also known as LKB1. And two things to point about these genes is that they're both known to regulate cellular metabolism in different ways. And for today's talk, I'm primarily going to focus on Keap1 and show you our work on understanding what Keap1 does, mutations do in lung cancer. However, I do want to point out that STK11 is also important. And the fact that these two mutations are so high, these two genes are so frequently mutated in lung cancer, suggests that metabolic rewiring through this genetic, these mutations and genetic adaptation in these tumors is very important. We think very important. So another thing to point out about Keap1 is that it's clonal. It's an early event. So this is a adaptation of one of the figures from the TRACER-X studies from Charles Swanton's group at UCL, where you can kind of see here the different mutations categorized as early drivers. So whether they're clonal, found in the majority of the tumor cells, as opposed to subclonal, as opposed to late mutations. Right? So as you can see, Keap1 along with KRAS, two genes that are co-mutated, are found very early. And along with 53 as well, and some other known genes that you might recognize are like BRAF, EGFR. So these mutations occur early and occur in the majority of the tumors, so suggesting that they may be important for tumorigenesis. What we understand now about these mutations is that they're clinically very, they're of major importance in the clinic. These are two survival plots here from looking at stage 4 patients from Memorial Sloan Kettering up the street here in New York. Showing that if you are KRAS Keap1 mutant, you do these patients do a lot worse than just KRAS mutant patients, as you can see on the left. And on the right, you can actually see the breakdown in terms of patients responding to checkpoint inhibitors, which is currently, along with chemotherapy, the standard of care in lung adenocarcinoma with KRAS mutations. So as you can see here on the right, if you are KRAS Keap1 mutant, you don't respond essentially to checkpoint inhibitors immunotherapy as compared to KRAS alone. Again, suggesting that studying these tumors is important because they remain a major clinical problem. So what does Keap1 do? So Keap1 binds to NRF2 and mediates its ubiquitination and proteasomal degradation through interactions with the COL3-RBX1 complex. What will happen is when cells accumulate various electrophiles or reactive oxygen species, among other stimuli that can actually regulate this pathway, you'll have dissociation of Keap1 and NRF2, and now NRF2 will accumulate and go to the nucleus and turn on a battery of genes, antioxidant genes, which I'll discuss in a minute. Again, and so what I want to point out is that along with Keap1, NRF2 as well is also mutated. So typically what you see in lung cancer and some other cancer types as well is that you see either loss of Keap1 or gain of function mutations of NRF2. So, and these are usually mutually exclusive. And what's important to note here is that 30% of all non-small cell lung cancer, which includes both squamous and adenocarcinoma, have mutations in this pathway. And as I mentioned, that there are mutations found in other cancers, either by point mutations or by copy number changes. And we do have evidence that, for example, NRF2 is required for tumorigenesis by a few studies, including one from Gina DeNicola's group, showing both in lung and pancreas KRAS-driven tumors that NRF2 is required, and from Masa Yamamoto's group. So as I mentioned, NRF2 is a transcription factor. So when it's activated, it turns on a battery of antioxidants. Essentially, it's a major regulator of the cellular antioxidant response. So it'll activate the expression of, it'll turn on the expression of genes that regulate iron and heme homeostasis, genes that actually promote NADPH production, an important reducing factor in cells. But it'll also induce the expression of antioxidants themselves. So glutathione, Isaac has very nicely introduced glutathione thyrodoxine during his presentation. These are two very important antioxidant factors in cells. Glutathione is a tripeptide, glutamate, cysteine, and glycine. Thyrodoxine is an encoded gene. And in addition to that, it'll also turn the multi-drug resistance associated proteins, which among many things, allows us to detoxify various drugs or oxidants that accumulate in cells and pump them out of the cells. And glutathione actually is one of the things that can also pump out of the cells as well, which is quite interesting. So again, it's a pleiotropic transcription factor. It activates a lot of different arms of the antioxidant pathway. So from what I've told you so far, this may seem counterintuitive, although from what we saw from Isaac's talk, this might make sense to you now. However, typically, it was thought that antioxidants suppress cancer, and that taking antioxidants can prevent tumorigenesis. However, there's been a battery of studies now, both in mice and humans, so clinical trials in humans showing that antioxidants led to a decreased survival in patients, or increased incidence of cancer, I should say. And several elegant studies in mouse models, including the mouse model I'll discuss today. And this is a study from Martin Berger's group performed by Volkan Sain here, showing that in KRAS mutant lung tumors in a mouse model here, dietary administration of N-acetylcysteine on the left or vitamin C on the right led to robust increase in tumor burden in these mouse models. Again, suggesting that dietary antioxidants promote tumorigenesis. In addition, there's been many other studies, including the study by Isaac that he presented last week, and several other studies showing that on one hand, antioxidants can promote cancer, on the other hand, oxidants can suppress tumorigenesis. So where does this fit sort of in our overall view of cancer metabolism? So this is a very simplistic way to view things, but as we know from studies a long time ago done by Otto Warburg, the so-called Warburg effect, we know that tumor cells potentially consume a lot of sugar to produce lactate. Now this model has been has been improved in many ways, because now we understand that tumor cells utilize oxygen, nitrogen, carbon, so forth in a very different way than normal cells. And this is in general to produce, the reason for this is to produce more biomass, to increase cell numbers, for allow cells to proliferate. So in order to produce nucleotides, amino acids, and lipids. So the idea is that while doing this, while producing more biomass, tumor cells actually may be producing more reactive oxygen species, which then in turn are cytotoxic even for tumor cells. They can damage DNA, they can damage RNA, they can damage protein. Okay, so of course, this is one end of the spectrum. The other end of the spectrum, and as a lot of people like Navdeep Chandel will tell you, is that reactive oxygen species, oxygen radicals, are also important for signaling. So oxidation of cysteines or potentially methionines in proteins may play a signaling role. So you sort of want to be somewhere in between, as Isaac kind of introduced. You don't want to have too much, but also too little may also be cytotoxic, toxic to cancer cells. So again, just going back to this pathway, what we wanted to really understand is, do Keap mutations impact the mechanisms of progression in lung cancer? Do they alter histology? Do they alter immune responses? And do they impact how these tumors respond to current standards of care? And can we then identify new therapies to specifically target these tumors? And this is sort of some of the things I'm going to focus on today in my presentation. So the way to go about this is, for us, was to basically take the information from patients, rapidly model these mutations in mice using CRISPR-Cas9 in vivo methods that we developed. And ultimately, once we just demonstrate whether Keap1 mutations are important for tumorigenesis, try and identify ways to target these tumors. So identify therapeutic vulnerabilities. And we do this by doing genetic screens, CRISPR screens, to identify so-called synthetic lethalities in the context of Keap1. And ultimately, we've established an array of both human and mouse preclinical models that allow us to test some of these novel therapeutic strategies and eventually take this into patients. And as I'll show you today, some of our findings that we've published in some ongoing work have led to current clinical trials in patients. So the model we use to study lung cancer was developed a long time ago in Tyler Jackson's lab. So this takes into account two different conditional CRE-LOXP conditional alleles. One of the so-called lock-stop-lox KRAS G12D, and the other one is a 53 flux allele. So the KRAS allele has a G12D mutation. And upstream of the G12D mutant exon of KRAS, there's a lock-stop-lox cassette that blocks the expression and essentially the production of this mutant version of KRAS. And in case of 53, most of the 53 gene is flanked with LOXP sites. So what we do is we use this lentiviral construct shown here above, which allows us to express CRE recombinase along with Cas9 driven by the same promoter. And in the same vector, we also have a guide RNA driven by a U6 promoter. So this construct, this lentiviral construct, now allows us to, in the same cells that are infected, express CRE recombinase stably, express Cas9 stably, and a guide RNA or multiple guide RNAs of interest against the genes that we want Cas9 to edit, to mutate. I should say this construct is available on Adgene for anyone that wants to try it, and it's been used now in several CRE-LOXP based mouse models, particularly cancer models, but it can theoretically be used for non-cancer CRE-LOXP models as well. So when we deliver, I should say, we make these viruses, the lentiviruses, we deliver them intratracheally in mice, so we make the mice inhale the virus, and not only in the lung epithelium we deliver the virus, and what that will do is that will activate KRAS, mutant KRAS, delete p53, and in the same time, in those same cells, stably introduce Cas9 along guide RNAs against genes we want to target. And the important thing about this model is that you get this very nice stage-specific progression from an early grade 1 neoplasia that eventually forms an adenoma grade 2, an adenocarcinoma grade 3, a grade 4 tumor that's highly desmoplastic and invasive, and eventually will metastasize. So you get a very nice stage-specific progression, similar to what we think happens in humans. So our question was, if we mutate Keap1 using guide RNAs, does this accelerate tumorigenesis? And indeed, we find that you can see on the top panel here is a guide RNase against a control TD tomato gene, so non-targeting guide RNA. You can see, you can actually kind of distinguish the tumors here in dark, grossly histologically here in dark purple. But on the bottom, you can see when you target Keap1, these tumors are a lot larger. When you look at them histologically by H&E, they look quite different, as you can tell. You don't have to be a pathologist. They're much more desmoplastic. And importantly, we can see now that in tumors where we've edited Keap1 in the bottom, we can see very high levels of NRF2 in the nucleus, as we would expect, and high levels of expression of its direct target and bona fide target NQ1, shown on the bottom right. So again, this is we can see that this was published a few years ago, so I'm not going to go into the details, but we can see that there's a really high tumor burden in these mice with Keap1 mutations. And what we see is that when we assess for a proxy of reactive oxygen species, we look at DNA damage in response to ROS, and you can see that the Keap1 mutant cells, the Keap1 mutant tumors have a lot less. And another thing we observed actually, which was not published originally, is that these tumors were actually quite metastatic. So even if you allowed the Keap1 wild type tumors to grow to the same size as the Keap1 mutant tumors, you still wouldn't get the same metastatic incidence as you do with Keap1 mutations, which was very interesting to us. And we wanted to investigate this further in collaboration with Michele Pagano's group at NYU. So as I said, it's a work in collaboration with Michele Pagano's group, spearheaded by Luca Lignito, a very talented postdoc in his lab, and worked together with Sarah Lebeff and Volkan Sahin, two postdocs in my lab, did a lot of this work. So what we did early on is that we took Keap mutant and wild type cells, and we did RNA-seq and proteomics, and looked for overlap between those two high throughput data sets. And what we found is that an enrichment of the BAC1 pathway. We found that the BAC1 pathway was enriched in Keap mutants tumors as compared to the Keap wild type tumors. We were able to validate this by Western blot. As you can see here on the right, you can see, so KP are Keap wild type, KPK are Keap mutant, and KPK plus Keap is Keap complementation. So in Keap null cells, putting Keap back in. And as you can see, as you expect, NRF2 goes up when you mutate Keap, it goes back down when you put Keap back in. But BAC1 follows the same sort of paradigm as NRF2. So does the very potent NRF2 target known as HO1, hemoxygenase 1, shown towards the bottom. So this was interesting to us because what was known in the literature at the time was that BAC1 and NRF2 were essentially antagonizing each other. So the literature suggested that when BAC1 is high, NRF2 is low, and when NRF2 is low, BAC1 is high, and vice versa. However, our data was showing something different. It was showing that somehow, either by Keap mutations or by activating NRF2 pharmacologically, by disrupting its interaction with Keap, we were getting high levels of BAC1 only at the protein level, I should say, not the RNA. And I showed you the protein data in a minute. So this was quite interesting to us. So we wanted to understand why this is. So we, through a lot of elegant biochemistry from Michele's group and Luca, we identified the mechanism by which this happens. I'm not going to go into a lot of the details. You can go read the paper. However, what we found is that NRF2 is known when it's activated by Keap mutations, it turns on constitutively HMOX 1 expression, hemoxygenase 1, which in turn will degrade free heme cells, which in turn will actually impact interaction of any protein that heme regulates. And what Michele's group found is that BAC1 is being degraded by FBXO22 only in the presence of free heme. So when you degrade free heme by hemoxygenase 1, BAC1 does not get degraded anymore, it becomes stabilized. And what's important about this is that BAC1 was actually known in the literature through studies mostly by Yi Kang's group in Princeton to be a pro-metastatic factor, at least in breast cancer. So we thought this was interesting. It could be that BAC1 through the stabilization, through this mechanism in Keap mutant tumors, could promote metastasis. And this is just to show you that this is a hemoxygenase dependent mechanism. So you can see here on the right, if we make clones that lack HO1, hemoxygenase 1, even if you activate NRF2 now, you do not get stabilization of BAC1, but you still get stabilization of NRF2. And of course, HO1 is not present. So you need HO1 activation, HO1 expression to be able to degrade, stabilize BAC1. And of course, we demonstrate in several different ways, just some of the experiments, that this is important for metastasis. So we found here on the left that if you knock out BAC1 with inducible shRNAs, you can completely suppress the metastatic potential of Keap mutant tumors. And importantly, if you use a pharmacological way, so using zinc protoporphyrin 9 to essentially inhibit hemoxygenase 1, you can see here on the right that you can actually suppress the metastatic potential of Keap mutant tumors. However, if you do the same thing in the context of FBXO22 loss, so if you lose the ubiquitin ligase that requires heme to degrade BAC1, now altering heme levels doesn't matter. So because FBXO22 is gone and BAC1 is always stabilized, really suggesting that the mechanism is quite linear, just like as we show it, as I demonstrated down in the bottom of the slide here. And we think this is interesting because there was also a parallel study by Martin Berger's group, nicely showing that in cases where you have dietary antioxidants, you have a very similar mechanism by which BAC1 can be stabilized and promote metastasis. So this can happen genetically by mutations, but this can also happen by dietary antioxidants in the diet. And we think this is also important. So we're very excited about this impact on heme that NRF2 may have. constitutively, because heme has a major role in a lot of physiological processes, including circadian rhythms, which is our lab is very interested in. It's known to play a role in various aspects of lipid metabolism. OXPHOS is important for the electron transport chain, and modulation of transcription of various transcription factors, including circadian transcription factors. So we're really interested in understanding whether some of these processes are actually altered in the context of Keap mutant tumors, or when NRF2 is stabilized by other mechanisms. So what I've shown so far is that we demonstrated that Keap mutations are important in lung cancer, and through the activation of NRF2, they allowed stage-specific progression, potentially early stages of tumorigenesis, but also enable cells to become more metastatic. So one interesting thing that we want to study in the lab, and I don't really have an answer, but we're really actively investigating, is, so as I showed you here, is that we think NRF2 overcomes reactive oxygen species. And Isaac nicely showed you that potentially in tumor progression, ROS can be very, very important, and ways by which cells can overcome it, for example, increased glutathione production, which NRF2 can actually do, can allow us to overcome these ROS bottlenecks. But where is the ROS coming from, and is it actually increased during tumorigenesis? So I don't think this is really well understood. However, there's a lot of elegant studies by Navdeep Sandel and others showing that low oxygen hypoxia essentially can lead paradoxically to increase of reactive oxygen species. And one of the hypotheses that we have is that potentially as tumor cells progress through different grades of tumors, larger tumors that become potentially more hypoxic, reactive oxygen species may increase. So this is something we want to understand. And we do have some evidence to at least show that despite the fact that they're in the lung, tumors that, these lung tumors do actually have hypoxic regions. So you can see here looking at hypoxic probe, an example on the bottom right of negative and positive tumors. You can see that the hypoxic probe staining is actually quite focal, which is we would expect. And you can see that if you go from early grade 1 and 2 tumors to grade 3 and 4, which are more aggressive and potentially more desmoplastic and more hypoxic, you can see that there's more hypoxic areas. So now we want to understand, are there more reactive oxygen species in these areas potentially produced, and do Keap mutations and NRF2 activation allows us to overcome these hypoxia-induced ROS? So in addition to understanding, okay, so what is the primary reason that NRF2 is required? Is it ROS? Is it metabolic rewiring? We want to identify ways to target it, right? So target tumors that have these mutations. So as I mentioned earlier, NRF2 is a very pleiotropic transcription factor. It has a lot of different targets. And being pleiotropic, we think that targeting individual nodes of the pathway will be redundant, cause redundancy. So because there'll be other ways by which cells can overcome, other NRF2 targets by which cells can overcome of targeting individual nodes. So our approach has been to identify dependencies that arise in response to the NRF2 antioxidant program being constitutively on. So the idea of this so-called reductive stress. So when you have high antioxidant production in cells, does that come with a cost? This high reductive potential of cells, does that come with a cost to other metabolic processes in the cell? And this can be thought as balance, shown here, where you have high antioxidant production. This will alter a lot of the sulfur metabolites and other metabolites, as I'll show you. So in order to produce a lot of antioxidants, but that will come with a cost to other metabolites that in the cell that maybe potentially allow us to target therapeutically. And there's been some work done on this already from some elegant work from Gina DeNicola's group showing, for example, how sulfur metabolism is different in Keap mutant tumors and how they can be targeted therapeutically. So we're not the only ones thinking about this. So our approach has been to take an unbiased approach, doing CRISPR-Cas9 based genetic screens. So we've, in collaboration with other groups, including Kivansh Brisso's group and Francisco Sanchez Rivera and Scott Lowe's group, we've taken the approach to generate targeted libraries against specific gene sets, so that we think are relevant in the context of this NRF2 Keap1 pathway. So we've generated libraries to target all the metabolic, known metabolic genes, ER stress pathways, proteotoxic stress pathways, for example. And the idea here is we use these screens to assess, can we identify genes that are required in the context of Keap mutations, but are not required if Keap mutations are not present. And this will give us an idea of genes that may be potential targets therapeutically, but also underlying, as I'll show you, biology ongoing in these Keap mutant cells. So, and I'm going to summarize some data that's already published here, where we performed such screens. So what we found in a study a few years ago now is that the transporter SLC1A5, known as a very important transporter for importing glutamine from the extracellular environment into cells, was synthetic lethal, found synthetic lethal in a CRISPR screen that we did in Keap mutant cells. So you can see on the bottom right here, validation of the screen. You can see on the right, if you Keap mutant, SLC1A5 loss causes growth arrest. If you're not Keap wild type, the cells don't care. And one thing I want to point out here is that SLC1A5 is expressed in both Keap mutant and wild type cells. So it's not that the wild type cells do not express 1A5 and therefore don't require it. They express it. They just don't require. If you lose it, they can overcome the loss of 1A5. Okay. So of course, glutamine when coming into cells is then converted to this enzyme called glutaminase to glutamate. And that was that's very important because there's already glutaminase inhibitors that are in clinical trials. And one of those is CB839. So we decided to test CB839, and we found here on the bottom left in vitro that if you test in multiple different Keap mutant cell lines, if you use CB839, it's very potent against these cells, are really low concentrations with minimal to no effect in wild type cells at these concentrations. Now, this was interesting to us. However, in prior work in collaboration, when I was still postdoc in collaboration with Sean Davidson and Matt Vanderhein's lab, we had shown that even though glutamine was very important for cell viability, for cell growth in vitro, when you went in vivo and either used CB839 or deleted glutaminase genetically using CRISPR-Cas9, it was not required for KRAS53 mutant tumorigenesis. So we thought, okay, maybe this in vitro data is an in vitro artifact. However, we found in multiple in vivo models, this is just an example here of some of the PDX models that we tested, that as we had shown before, again, in Matt Vanderhein's lab has shown before, if you are KRAS53 mutant alone in black, the tumor cells don't care, they do not respond to CB829. But if you have in blue Keap mutations, now you respond to glutaminase. This is a very potent effect of glutaminase when you have Keap mutations, and we've shown this now in multiple different animal models, both human and mouse models. Importantly, there's been several other studies since ours demonstrating similar data. I actually also forgot to include ones from John Hamack's group in MD Anderson. However, in these studies here in different models, pancreatic models, breast cancer models, or they've shown that NRF2 activation in different contexts causes dependency on exogenous glutamine and sensitivity to CB829. Again, supporting sort of our hypothesis that if you have Keap mutations or NRF2 activation, cells become sensitive to, dependent on glutamine and become sensitive to glutaminase inhibition. And this has led to a phase 2 trial sponsored by the NIH now, that will specifically test in patients that have Keap mutations or NRF2, whether glutaminase is effective against these, glutaminase inhibitors are effective against these patients. So very soon we're going to find out whether this pans out in humans too. We're very excited to see the results of this clinical trial. Now, what I didn't tell you though is why are these tumors more sensitive to, more dependent on exogenous glutamine? So what we found is that Keap mutant tumor cells on the bottom left here actually consume a lot more glutamine. That's something that we sort of expected. If they rely more on the SLC1A5 transporter, they rely on glutaminolysis, they probably secrete more glutamine. Uptake more glutamine from the microenvironment. However, what surprised us was when we looked at intracellular glutamate, we noticed that the Keap mutant tumors and tumor cells in blue had a lot less intracellular glutamate, and a lot of it was actually secreted to the extracellular space. And this was interesting because this got our attention because there was actually evidence showing here on the left that this antiporter known as System XC or XCT, that consists of SLC711 and SLC3A2, known to exchange glutamate for cysteine. So this is a mechanism by which a lot of cancer cells actually obtain cysteine. They obtain it in the form of cysteine from the microenvironment. So cysteine is two cysteines together. And in order to uptake a molecule of cysteine, they have to export a molecule of glutamate. So what's also important to note is that SLC711, which is one of the components of the antiporter XCT, along with SLC3A2. SLC3A2 is a strong target of MYC, so it's usually upregulated in most cancer cells, and it's found as a co-transporter for other antiporters in cells as well, not just XCT. But SLC711 is a direct target of NRF2, and as you can see on the upper right here, it's very highly induced in Keap mutant cells, and it's known to be a strong target of NRF2. So we asked the question, could the dependency of cells on glutamine and the efficacy of glutaminase inhibition, so use of CBA29, be because cells are constantly exporting glutamate outside of the cell in order to uptake cysteine? And could this be the mechanism by which cells are sensitive to glutaminase inhibition? Indeed, that's what we saw. If we inhibit pharmacologically XCT using a drug called Rastin, or we did this genetically, you can see on the bottom right here in green that treatment with a Rastin completely desensitizes these cells to the glutaminase inhibition. Again, suggesting if you block the export of glutamate from cells, increase the intercellular glutamate levels, now these cells do not respond to glutaminase inhibition anymore. They are resistant, just like the wild type cells in the left. I want to point out this mechanism was also supported by data by two other groups, Boy-Gann's group at MD Anderson, and also Matt Vanderheinen's group and work done from Alex Moore, who's going to be presenting. These are the citations on the bottom left, who's going to be presenting in this seminar series later on. So just to summarize the mechanisms, we think that by NRF2 activation, by multiple different mechanisms, promotes glutamate export from cells. And this now glutamate becomes rate limiting the ability of cells, for example, to feed TCA cycle, as we and others showed, because glutamate is a very important source of carbon for the TCA cycle. But what also is important to note is that glutamate is not just an important source of carbon for TCA cycle. It's not just used as an exchange factor to bring in cysteine into cells. It's also the major donor for the nitrogen group that goes on, the amino group that goes onto most of the non-essential amino acids. So on the bottom here, you can see an example of a transamination reaction. And you can see that the amino group comes from glutamate in these transamination reactions on the amino acids. There's multiple amino acids that are synthesized using glutamate as a factor here. And this is showing some of them. Serine synthesis, aspartate synthesis, alanine synthesis require glutamate. So we asked the question, if glutamate in the context of NRF2 activation and Keap mutations is constantly being exported from cells, does this make its availability rate limiting for non-essential amino acid synthesis? And does that alter the way by which these cancer cells obtain their non-essential amino acids? So this is work done by Sarah LeBeff and Warren Wu, and a postdoc and a very talented MD PhD student in the lab. So what they found is that if you look at cells in culture, you observe that in general, Keap mutant cells uptake more of the non-essential amino acids from the media. Uptake is showing towards upwards direction, secretion towards the bottom, with the exception of glutamate, which of course I already mentioned to you is highly exported from Keap mutant cells. And I want to point out that we didn't just see this in these cell lines, we also saw this in multiple other mouse and human cell lines that we actually tested. So we think it's quite general phenomenon in Keap mutant and NRF2 mutant cancer cells. So importantly, these amino acids are not just uptaken by cells; they're also important for their viability. So if you remove asparagine, remove glycine, remove serine from the media of these cells, you can see that the Keap mutant cells will arrest, as compared to the wild type cells that do not care; they'll keep growing just fine. And importantly, if we use a enzymatic method to degrade asparagine in the media using asparaginase, a drug used to treat childhood leukemia since the 60s. You can see that we can have a potent effect on using low levels of asparaginase in mutants, Keap mutant cells, opposed to wild type. But what's important to note here is that asparaginase can degrade glutamine, but in all the conditions in vitro and in vivo that I'll show you later, we've actually demonstrated that there's no effect on glutamine. We're just degrading asparagine. So we don't think this is just a specific effect in lung cancer cells. So we got some KRAS53 mutant pancreatic ductal adenocarcinoma cells from Alec Kimmelman's group. And we actually showed that if you activate NRF2 pharmacologically in blue, you can see now that you sensitize the cells both to serine and very low levels of asparaginase in vitro. Again, suggesting that you can activate NRF2 and program cells into the state that basically now makes them dependent on exogenous non-essential amino acids. So just to sort of demonstrate some of the mechanistic understanding of why this happens. So of course, our hypothesis is that glutamate is exported by cells to XCT actively in order to bring a cysteine. And now this makes glutamate rate limiting for the transamination reactions. So one of these reactions is mediated through PSAT, which enables synthesis of serine from glucose. And what you can see here, as I showed you before, that if you remove serine from the media of Keap mutant cells in blue, you can see that they arrest. However, if you actually now block the glutamate expo by using a rastin, you can completely desensitize the cells shown on the far right here to serine deprivation. And you can do the same thing if you actually increase glutamate. If you increase glutamate levels extracellularly, this will alter the gradient of cells of glutamate and cysteine extracellularly, intracellularly, and it will essentially block similar to what a rastin does, and you can see the same effect. Both glutamate and a rastin treatment completely abrogate the sensitivity of cells to serine deprivation. And the other sort of amino acid that I mentioned to you before, which we think is important, is asparagine. And we find the same thing here too, that if you actually supplement glutamate in the media or treat cells with a rastin, you completely bypass the sensitivity to asparagine deprivation. Now, of course, this is all in vitro data where there's a lot of the microenvironment, the oxygen tension is different. There's a lot of differences in vitro versus in vivo. So we really wanted to understand whether this is relevant in vivo. So to do this, we got ideas from Karen Vausen's group and Matt Vanderhaan's group, who've shown in the past that you can actually modulate serine and glycine in the circulation and their availability by using a serine and glycine diet. And on the left here, what you can see is that we let the mice establish tumors, then we put them into a control diet that has all amino acids versus one that lacks serine and glycine. And what we can see is that putting Keap mutant tumors in a serine and glycine deprived diet in dashed blue line suppresses their growth. In fact, now they grow at the same rate as the wild type tumors. And this has no impact at all in Keap wild type tumors. And this is interesting because previous data from Karen Vausen's group had actually shown that KRAS53 mutant tumor cells do not care about serine and glycine deprivation in vitro and in vivo, which is what we see too. But now if you put if you mutate Keap or activate NRF2, now you see that these cells become sensitive to serine glycine deprivation. So the mutation context really, really matters. Okay, just having KRAS53 alone does not necessarily define all human KRAS mutant tumors. There's a lot of mutations that occur along with KRAS that can define their metabolic dependencies. Now, we do the same thing with asparagine. We can deplete it in the diet. This is not very effective. It will limit asparagine levels, and same thing for serine and glycine. I didn't show the data, but we can bring down the levels in the diet by about 50%. But you can see, despite that, you can see some effect on tumor growth only in the Keap mutations. However, when you use asparaginase, you can see here recombinant asparaginase, we can see a very robust effect in the growth of the Keap mutant tumors and a little bit of an effect also in the wild type tumors. Again, showing that this is a very robust genotype specific requirement for exogenous asparagine, same thing as what we see for serine and glycine. So our model is that basically is that glutamate export, and this can happen in many different ways. In our case, this happens in response to NRF2 activation, becomes rate limiting for non-essential amino acid synthesis and makes cells dependent on exogenous amino acids. So we decided to test whether we could combine CB829 treatment along with depriving some of these amino acids. And you can see on the top right here, if we take Keap mutant tumors, we can actually show that serine and glycine deprivation along with CB829 have a very similar effect. And if you combine them, there's a little bit of an additive effect, nothing dramatic. But our surprise was when we did the same thing in Keap wild type tumors, where we saw that, as was shown before by others and us, serine and glycine deprivation alone has no effect. CB829 alone has no effect. But now if you combine the two, you see very strong synergy. Again, suggesting that limiting glutamate availability, not by exporting it, by limiting its production through glutaminase from glutamine, now sensitizes cells to the deprivation of exogenous amino acids. So what I've shown you so far is that through rational approaches, we've been able to, using either diets, recombinant enzymes that degrade amino acids, or compounds that are in phase 1 trials, we have taken a multi approach to target these Keap mutant tumors. So, and if you visualize again back in the balance that I showed you earlier, we think that Keap mutations or chronic ROS exposure will increase antioxidant production in cancer cells, produce more antioxidants, alter sulfur metabolism, but that will decrease the availability, which can impact on essential amino acid synthesis. It can impact on aplerosis, so TCA cycle feeding, and this can be therapeutically targeted by some of the approaches that I showed you today. So this work, the latest stuff I presented was published about a little over a month ago in Cell Metabolism. This was the cover in the journal from that month, showing Achilles and NRF2 being like a shield protecting him from ROS and chemotherapy potentially. And however, the Achilles heel being like targeting different metabolic vulnerabilities in cells that have high NRF2 through Keap mutations. So towards the end, last part of my talk, I just want to discuss sort of our studies on understanding whether mutations in the tumor and genetic subtypes of cancer that alter metabolism in the tumors, how they may impact the microenvironment. So how they may impact the metabolic milieu in the microenvironment, and how that metabolic milieu in the microenvironment can have an effect on cells in this microenvironment, including, for example, as shown here, effector T cells. And this is work done by Anastasia. Most of what I showed today is done by Anastasia Zavitsan and Ray Pillai, who are co-mentored by Sergei Korolov's lab. Anastasia is a graduate student, and Ray Pillai is a medical fellow in the lab. He's a pulmonary fellow. I want to give a shout out to Ray because he's been working 12-hour days every day in the Brooklyn hospital treating COVID patients, putting himself in danger. He's a real hero, and he's been an amazing person in the lab, and I really hope he's doing well during these tough times. So to put this in perspective, so our idea is that potentially having certain mutations in the tumor can be shown here in like tumor cells in green or red or gray. Some of these mutations in response to adaptation to nutrient availability, to hypoxia, or to ROS may be selected for. So for example, here I'm showing in red Keap mutant cells. However, we think that maybe in addition to being selected for the tumor cell autonomous effects, these mutations may also be impacting the microenvironment. So impacting the infiltration of shown here T cells, various myeloid cells, or other immune cell types, either innate or adaptive, which can potentially impact the infiltration of these cells and their function. So we think there's already some existing data in the literature showing that these mutations may have an effect on the immune microenvironment. So in the bottom here, this is a paper from a year and a half ago now, where the major conclusion of the paper was if you have high tumor mutational burden, these tumors are more likely to respond to immunotherapy. There's many studies have shown this now. However, in their last figure, they looked at whether mutations in the tumors, so sort of frequently occurring mutations, associated with the poor T cell infiltration score, shown here as what they call GEP. So this is a lymphocyte infiltration score that's calculated by bulk tumor RNA-seq, where basically you, and methods such as CyberSort, which you basically look for T cell signatures within the bulk of the tumor by RNA-seq. And what they found is that if you have Keap1 mutations in both lung squamous and lung adenocarcinoma, these tumors are associated with the poor lymphocyte infiltration score. And the same thing is seen if you have STK11, LKB1 mutations. So again, this suggests that these two metabolic mutations, which I introduced to you today, and importantly, Keap1, may be somehow impacting the infiltrations of T cells. And as I showed you earlier, in clinical trials, do not have any suppressed ability of tumors to respond to checkpoint inhibitors, immunotherapy, immune-based agents. So how does this happen? How can we, the idea is, does Keap1 metabolic rewiring do this, exclude T cells? And can we reverse this using metabolic strategies in combination with immune-based therapies? So to do this, we use our genetically engineered mouse model. We use a slightly different version of what I showed you before, in which case now the Cas9 is not delivered with a lentivirus, is delivered, is found in the mouse, in the ROSA locus, lock-stop-locks Cas9, so it's also pre-dependent. Across these mice to KRAS conditional p53 conditional mice. And now what we do is we can deliver either guide RNA against neomycin or a non-targeting guide RNA, or guide RNA against Keap1 along with expressing cre recombinase. We can deliver these viruses in the lungs, initiate tumors, and look at, okay, what happens to tumor burden? We know Keap1 accelerates tumorigenesis. But importantly, in this context, we want to understand how do Keap1 mutations impact immune cell infiltration? So we do this by multicolor FACS, looking at adaptive and immune cells. By multicolor IF, 6 or 7 panel immunohistochemistry, both in mouse and human tumors, or more new methods that allow us to do up to 30 or 40 markers such as Codex, and again, to visualize where the immune cells are in the tumor. But also we can do single cell RNA sequencing, of course, to give us a granular idea of like what is going on in terms of the immune cell population in these tumors. So what we've seen so far is that when looking at total T cell infiltration, you can see it's dramatically lower in the left here in our mouse model tumors. And this is, I should mention, regardless of tumor size. So you might say, okay, Keap1 mutant tumors are larger. Potentially they can cause T cell exclusion. However, what we see that even in smaller tumors earlier on, if you look at early stage disease, you still see the same thing. You still see that grade 1 or grade 2 tumors that are not that desmoplastic, they're not known to exclude T cells, they still have a lawless T cells if they're Keap1 mutant as opposed to wild type tumors, which is an important distinction to make here. And on the right, we've also sequenced tumors at NYU, and our pathologist here, Andrea Moreira, scored them for T cell infiltration. And you can see here that in general, if you're mutant on the right, you have less T cell infiltration as opposed to wild type on the left. And we're increasing our numbers here. We can visualize the T cells in the tumors. This is both in mouse and human. These are human tumors. On the top is a wild type tumor. On the bottom is a mutant tumor. As you can see immediately, the mutant tumors have a lot less T cells in them, and indicated here by green CD3 T cells. But they have a lot less CD8 T cells as well. We can also quantify FOXP3 positive cells. These will be CD4 positive, FOXP3 positive Tregs. The PD-1 checkpoint inhibitor and pancreatin marks the tumor cells. And we can do quantification. You can see here on the right that if you're Keap1 mutant, you have lawless T cells in general, as I showed you also in the mouse data and by IHC in human tumors. But also you have less CD4 T cells and CD8s. Again, strongly supporting some of the computationally performed CyberSort data from human tumors using our bulk RNA-seq. We can actually, in our case, visualize this in the tumors and do better characterization of these immune cells. And what we can see here on the top is that when we do FACS from the mouse tumors, we can actually characterize the immune infiltrate. We can see in general as T cells. A trend towards less CD4 and CD8s. But importantly, not the numbers. It's not just the numbers that decrease, but what we see is that the CD8 T cells, the cytotoxic CD8 T cells that are in the tumors, are less active as well. Okay, so they express less TNF alpha, less interferon gamma, less IL-2. These are important cytokines, chemokines required for T cell important for T cell activity. Of course, we want to really get a better understanding of this immune microenvironment, and we think single cell analyses, of course, are great to do this, because you can identify not just changes in the numbers of certain immune cells, but also the transcriptional programs and potentially their differentiation status. This is an experiment that was done relatively recently in our mouse model, where you can see we can identify many different clusters. I've annotated here different types of macrophages that we can identify. Lungs are very interesting because they have resident alveolar macrophages, which are non-bone marrow derived, but there's also bone marrow derived ones that can come, monocytes that can come to the lungs, and in the context of the tumor now differentiate. And we want to look at those too. And of course, as you can see, we can also detect a lot of neutrophils, T cells and B cells. So now doing these RNA-seq analysis, we're trying to get an idea of like, oh, do we validate what we see by FACS, the decreased cell numbers of some of these immune cells, decreased activation markers, but also do we see particular differences in their differentiation, okay, which this method can really help you identify. So as I showed you today, we think that Keap is important in promoting many different stage-specific aspects of tumorigenesis. So we think Keap through NRF2 activation can promote tumor progression by suppressing ROS in tumor cells, allowing them to overcome this bottleneck. As I showed you, we know that through a redox mechanism, post-translation mechanism, NRF2 can actually stabilize BAC1 and promote metastasis, but it can also promote immune evasion, right, through mechanisms that we're still characterizing. And as I showed you today, we've made some advances in terms of identifying different therapies to target these Keap mutant tumors at various stages of disease progression. So I showed you that HMOX inhibition, assuming there was an inhibitor, could potentially be used as a way to suppress the metastatic potential of these tumors. But I also showed you through either glutaminase inhibition or amino acid restriction through dietary intervention or enzymatic ways to limit amino acid availability, we can suppress tumor growth at earlier stages of disease progression in these tumors. And ultimately, we think we're modeling a genetic subset of lung cancer. That's very important. However, what's important to note is that Keap mutations do not occur in isolation. This is going back to the sort of evolution of human lung adenocarcinoma tumors, right? So, and these are just mutations. These don't have to happen in the same tumor. However, this is categorizing mutations in patients in different early versus clonal, subclonal versus late mutations, very clonal mutations. Subclonal mutations. But what we know from these studies is that Keap mutations can actually co-occur with other mutations that happen later that are subclonal. For example, one of those could be STK11 or another gene called SMARCA4 and other ones. So it may be that what we're modeling in our mouse is an earlier stage of the disease, which is important to know and understand. However, tumors evolve, and we want to be able to understand how they evolve and why the different mutations allow them to evolve. And is this metabolic? Is it immune? Etc. So with that, I'll end this presentation, and I'd like to thank my lab. I already acknowledge most of the people at this work. This work would not be possible without our funding sources on the bottom left here, and of course our collaborators who have been instrumental in these studies. I already acknowledged the ones that were involved in some of the work that I showed you today. And thank you for your attention. I look forward to answering any questions you may have or on Twitter. Yeah, so thank you all, and stay healthy and safe.